All right. So yesterday, I apologize. The notes were improperly printed, and as a result, you only had half the pictures. And I've also now realized that I should not use certain copiers in the copy room, because now, as you're looking at it, I realize that the drawings are actually completely black. So that's not helpful either.
Don't print them again. I guess we can't take notes today.
Good news is we have it here. If you want to follow along with your Chromebook, you can. Please don't play Fortnite. You can also follow along with the book, because these are taken from the book. So if you've got an online copy or the real copy of the book, you can use that too. Okay. I have now started marking down which printers not to use in the copy room. Yeah. Because I keep going in there and don't even get me started. Okay. Anyway, yesterday, we were talking. I'll take it away from you. Be nice. Okay. We were talking about the start of the unit, gene expression. Gene expression is taking our DNA, taking our genes, which are recipes, and taking those recipes and turning them into a phenotype. So we talked about in genetics about phenotypes, what you look like, what traits you have, what diseases you're more likely to get or less likely to get, or what immunities are you likely to have or less likely to have. That's your phenotype. So we have to figure out how to convert our genetic information into a phenotype, what you actually look like. And we talked about how that happens in two steps if you're talking about bacteria, which again are the eukaryotes or the prokaryotes? Prokaryotes. I'm going to start trying to throw a little bit more at you, making you kind of dredge up, get rid of the cobwebs for some of the stuff from earlier units, because AP test is coming, so we need to start, thinking about that. So I'll start throwing stuff in and making you think back to previous units. So this is a prokaryotic cell. It has transcription and it has translation. Okay, two parts. Gene expression, two parts. If we look at a eukaryotic cell is actually going to have three steps to it. It's going to have this transcription. And what do prokaryotic cells not have? They do not have a nucleus. So here we have a nucleus. And this is because we have a eukaryotic cells. We have organelles. Okay. You got one, Sophia? Do you have the new one? Yeah. Okay. Just checking. So transcription's the first part. The second part is going to be some processing. We're going to talk about that processing today. And then the last part is going to be that second step that we see in prokaryotic cells. So there's third step here because we're in a eukaryotic cell. Translation. Okay. So those are our three steps that we're going to have in our eukaryotic cells. We're going to focus mainly on eukaryotic cells because they are a little more complicated, but in a good way, in a way that makes us have more flexibility with our genes than prokaryotic cells. And I think, remember where we kind of left off here. I'm going to read some of this and skip a little bit of it. Okay, so when gene expression happens, how many strands of DNA do we have in a DNA molecule? 1, 2, 5. How many strands are there? 2. Oh, wow. We gotta really get those cobwebs out. Yeah, we have 2 strands, right? Right? Yeah, one matches with the other, right? We have A's with T's and C's with G's. Okay. So, when we do gene expression, we're only going to use one of those strands, and that strand is going to be called the template strand. Okay, it's going to be called the template strand. And that is the one that our mRNA is going to work off of. Okay. So the template strand is always the same strand for the given gene. Then there's the other strand that evidently I'm not going to talk about yet. Hold on, let me just go forward. No, maybe not. All right, never mind. I'll come back to that one in a second. As mRNA does its job, it is going to read what we call codons. And those codons are 3 letters, 3 bases. So an A and a C and a G, or a G and a C and a G, or a G and a G and a G. Okay, so they're always 3. They're 3 bases. They're 3 nucleotides. And they're always read in the 5 to 3 direction. So remember when we talked about DNA replication Gesundheit before, we talked about how these enzymes have a directionality. They have to go in one direction, and that's the 5 to 3 direction. Okay. Oh, that's what I wanted. The non-template strand. So the one that mRNA is not reading off of is called the codon, or sorry, the coding strand. That would probably be better if that was bolded. Okay. Let's just bold it. There we go. Because it is going to be exactly the same as the mRNA, because the mRNA is making a copy, or it's making a complementary copy of the template strand. So the coding strand and the mRNA strand should be identical, except for what? T and U. Good. So the T is going to be replaced with our U. Okay, because RNA does not have thymine, it only has uracil. Each of those 3 triplets, 3-letter codes, will code for a specific amino acid, and that will serve as part of the growing chain of protein that we're going to be making. We'll talk more about that as we get into this. So I alluded to this earlier yesterday, that is, and I said that there were 20 amino acids. So we have 4 bases, A, C, T, and G. That's it. That's all we have. So if I have just A, C, T, and G, can I make 20 amino acids? Well, I can't have 1 letter, right? If I had 1 letter, how many amino acids could I make? 4, right? I could have adenine, thymine, cytosine, and guanine would each code for an amino acid, but I need to get to 20. Okay. Yeah. So the answer is 3. You need to have 3 letters in order to have enough combinations to make 20 amino acids. Okay. And this was figured out. It's not really, I just wanted you to think it through, but you are always going to be given, if you need it, you'll be given this thing, which should look familiar. Have you seen this before?
Nope. No.
Really?
I think so.
Boy. All right. So this is a codon chart. We'll talk more about this when we get to the 3rd part of gene expression translation. But there are, let's see if I can count them up here. All of these 3-letter words are different amino acids. So they should add up to, some of these I think I'm circling more than once. They should add up to 20. Okay. But you can see that some of these, most of them, have more than 1 code, right? So, for example, proline could be CCU, it could be CCC, it could be CCA, it could be CCG. Okay, so it can be 4 different codes. right? You look confuzzled. Are you confuzzled? No? Okay. Just checking. So this was discovered back in the day, that there are 61 different codes that code for amino acids. 3 of those codes, we call them triplets sometimes, codons or triplets, are stop. So they say, stop, you're done. You're done. Stop, stop. What you're doing? Stop, stop. I do that with my freshmen a lot. Stop, stop what you're doing. The genetic code is redundant. More than one codon may specify a particular amino acid. No codon specifies for more than one amino acid. So if each triplet code, whatever it is, only codes for one amino acid. Codons have to be read in the correct grouping, or what they call reading frame. And what that means is, if I have that's supposed to be 3 C's, and say I have a U there. First 3 letters are what? C, C, C. Sometimes there was what we call a reading frame error. And that could be because one of those letters drops out. So now what is it? CCU. So let's look at our chart here. Raggy. Can we erase this? There we go. Are they the same? Damn it. Sorry. Let's try a different one then. I shouldn't have used proline because proline is always the same. Well, let's look here because it'll be easier. Okay, if I had CAG, what is CAG code for? There it is. Oh, that'll work. And I changed it to, let's say I have a U in there too. First 3 letters would be CAG, right? But now I take that out, it's AGU. What happens now? What did I change it to? Yeah, I'm going to change the amino acid from that to this. Now, that can be devastating because it will produce an amino acid. Oh boy, we're going to go back to unit 2. Oh no. Let's pretend that this amino acid is hydrophobic and this one's hydrophilic. What's going to happen to the overall shape of the protein? Yeah. I mean, the short of it version of it is going to be that it's going to be messed up because why? What is this sear going to do that this Q wasn't going to do? If this is hydrophilic now, philic, it likes water. It's going to mix with water, so it's going to change its location. So instead of being like in the middle of, let's say that's the amino acid, and I got a sequence of a whole bunch of amino acids, right? It's in the middle there, but now all of a sudden when it becomes hydrophilic, it's going to move it. So now all of a sudden this guy's on the outside. That changes the overall shape of the protein. If it changes the shape of the protein, it's not going to work the same way because why is shape of the protein important? It's part of its function, yeah. So if it doesn't have the right shape, it's not going to function correctly, right? Just like we talked about enzymes And enzymes have a special three-dimensional structure. If you change that three-dimensional structure, you're going to change what the enzyme can do. It's not going to work. What do we call it when an enzyme doesn't work anymore? Denature. Good. See, you all know that. We just need to kind of scrape away stuff, right? Okay, so scrape away some cobwebs. So the cool thing about this, and we'll learn more about this as we go through this unit. One of the things we're going to do is, well, I'll tell you in a second. One of the cool things about this unit is the idea that every living thing on the planet, from the most simple bacteria that you have to, slugs, to the cockroaches in the back, to little Charlie the mouse, who's now going back and forth in my room, to us, we all have the same ACs, Ts, and Gs. Our DNA is the same, except for the codes. So the codes are different. So different proteins will have different shapes, they'll look different, and they'll produce different outcomes. But they all start with those same 4 letters. Okay? Because they start with all those same 4 letters, what we can do is we can theoretically, and not so much theoretically anymore, we can actually do this, is we can take a gene from one species, and we can take it from that species, and we can put it into another one. And I'm sure this one turned out as, you need more water? Yeah. Go ahead, right back there. Is you can take a gene from a firefly and you can put it in a tobacco plant. You can make it look fluorescent. Even more scary, you can take a jellyfish gene that makes that jellyfish fluoresce in the dark, and you can make his snout and his hooves glow as well. Because it's fun? No. There's a better reason. Yeah.
Like lizards can regrow tails. Okay. We take that and make it, we can regrow limbs.
Ooh, I like the way you think. Well done. In theory we could. In theory we could. I'm sure I'm not a developmental biologist, so I'm sure there's lots of other steps that would have to go in there. But yeah, so these were done for specific reasons, but these are genetically modified organisms. You've heard that word before, right? Yeah. Mira, did you have a question? Yeah, so when we get to development, we'll actually talk a little bit more about how there are some parts of us that are inactivated, and if you could turn them on, we could probably get some roach-like areas. Yeah. That's what's so cool about science fiction, is it's when it's done well, it's based on science. So that would be harder. Because they wouldn't have that genetic material in there. Yeah. Good question.
Yeah, wait.
Yes.
Why like tobacco? Why just like feel like
Well, I'm not sure about this one. My understanding about this guy, the pig, is that putting on my farmer hat here. Actually, I never raised pigs, but when you take a hog to market, you want that hog to be as big as possible because they pay based on the weight. There's so much per pound. Okay, so you want it to be as big as possible. So they've done all sorts of breeding for a long time where they're just trying to get a bigger hog, right? Well, they did this experiment where they actually took genes from hogs that produced a bigger muscle mass, and they put it into, I don't even know, they put it into other embryos to get this bigger pig. But the thing is, you don't know when it's a little piglet, you don't know if it's going to grow up to be one of the bigger ones or not. So they put in a visible marker, and that visible marker was this fluorescent gene. So they put in a gene for muscle mass, and they also put in a gene for the fluorescence, so that when those were expressed, gene expression, when they were expressed, you'd have a marker when they were young to say, yeah, this is one of the guys that's going to get really big. Sophia. Well, they have artificially made it a linked gene. Yeah. Good point. Actually, that's an excellent point.
Could a pig still get the muscle mass without fluorescence? Or could it just fluoresce?
I think I'd have to look at the actual procedure, but my guess is they're doing it after meiosis has occurred, so that crossing over wouldn't take place. Because that's kind of the whole point. They want to insert it in a way that they'll get the max number of hogs that will have that muscle mass. But yeah, if you did it before the sex cells were formed, then yeah, totally could happen. Anyway, we are going to do an experiment where we take jellyfish DNA and we're going to insert it into a bacteria and we're going to make it fluoresce. So using a UV light, we'll be able to see these bacteria that have picked up the genes that we're interested in. Yeah, Maren. We could theoretically. That would be fun, wouldn't it? So you can in the pet store, and this has been a long time ago. I actually got fluorescent fish. Have you seen them? Glowfish. Yeah, they have genes from other species inserted into them. Kind of cool. You could, yeah. And that brings up the whole ethical thing. Should you?
Wait, so like you can't do it when they're alive?
I was thinking maybe Lucas would want to do it. I mean, he's got the hair for it. All right. So let's keep going. So first stage in gene expression is called transcription. So we're going to take a little bit closer look at that. Remember DNA replication? Ugh. What was the enzyme that we used to start the whole process? Helicase we used absolutely first. You're right. I was thinking about when we were actually going to start making what kind of polymerase? RNA polymerase. Yeah, good. It's right there. Yeah, that's why I took it off the page. So RNA polymerase is going to start this whole process because we're not making DNA now, so we don't need an RNA primer. We're actually trying to make RNA. Okay, so we're going to use this RNA polymerase. And Grace, what you said about helicase is right, but RNA polymerase actually can do double duty. It can do other things. And one of the things that it can do is it can open up, it can separate the DNA strands, it can break apart the hydrogen bonds that are holding those nitrogen bases together. Okay, so it can do the job of DNA helicase. It doesn't do that in DNA replication, but it does do it in gene expression. There is no helicase in gene expression, in transcription. So RNA polymerase is going to work to produce RNA molecules, to produce our mRNA. So RNA polymerase doesn't need a primer like DNA did. And it's going to follow the same rules as the rules we had before for base pairs, except for we know that thymine doesn't exist in RNA, so we're going to substitute that with uracil. Okay, so we're going to have a U instead of T's. This next page is showing kind of the whole setup, and I'm going to go through it in more detail, so I'm not going to spend too much time on that. One of the things that the RNA polymerase needs to know is where to start. And where to start is an area called the promoter. Okay, the promoter. In bacteria, there's a sequence at the end called the terminator. There's a different way of doing it in eukaryotes. And then the stretch of DNA that's being actively copied is going to be called the transcription unit. Okay. There's going to be 3 parts to it. There's going to be, they're very straightforward. Initiation, so getting it ready to make a copy or make a transcript of it, actually doing it, the elongation, and then the termination when you're like, stop, we're done. Okay. Those are going to be the 3 parts. We're going to talk about promoters. That promoter area, that is a sequence of nucleotides. So A's, T's, and G's, because it's all just DNA. But it's a sequence that's going to tell the RNA polymerase where to start. Okay, where to start. And it is what we call upstream of the actual place where you need to start making a copy of the DNA with mRNA. So upstream just means it's before it. Okay, and then you can also talk about downstream, which is beyond it. It's just a term that is commonly used. There are also these things called transcription factors that are either necessary or helpful, depending on the situation, to allow the RNA polymerase to bind. You can kind of think of it as they actually build kind of like a scaffold, a little structure that allows the RNA polymerase to attach and not fall off of the DNA. Because one of the things that can happen is RNA polymerase, because it's kind of sitting on that DNA, kind of like a train on tracks, it can fall off if it doesn't have some help to kind of stabilize it and make it stay where it needs to stay. So those transcription factors need to be made. And you can actually regulate whether or not gene expression happens by either causing those transcription factors to appear or inhibit those transcription factors so they don't appear. So it's a way of kind of fine-tuning the whole process. The whole complex where you've got the transcription factors and the RNA polymerase bound to the promoter, all together, that is called the transcription initiation complex. Now, this is where I always get the most laughing in this unit, I think, probably. The promoter consists of a sequence of repeating TA, TA. So it's sometimes called the TATA box. TATA box. Okay. And that is really important because if the RNA polymerase doesn't see that TATA, TATA region, it's kind of like an airplane trying to land. If you can't see the lights, it's just going to keep going. It's like, I don't know where to land. I'm going to take off again. Maybe they'll replace the bulbs. I don't know. Something's going on, right? So same thing here. RNA polymerase cannot land unless it has that promoter sequence. If it doesn't have that TAA, it will not be able to transcribe the DNA. If it can't transcribe the DNA, it can't make a copy of it. If it can't make a copy of it, it cannot do gene expression. Okay, so that's a very important sequence. Let's see. What do I got here? Okay. Top piece here is showing you the DNA. So we're upstream here. And here's my TATA box. TAAA. And then I have my non-template or my coding strand. Okay. So it could be the top, it could be the bottom, depending on the enzyme that you're working with. It just depends. And also, obviously, the direction. So it's always got to go from 5 to 3. And then here's my little bubbles, which are my transcription factors that help hold my RNA polymerase in place. So the RNA polymerase is going to recognize that TATA box, and it's going to land in that area that's just upstream. This is where we're going to actually start, right here. is where we're going to start transcribing. But it has to land before that so it can start its job at the very first letter of the DNA. This thing that, who plays guitar in here? Somebody plays guitar in here, don't they? Yeah. So what does that look like to you? No, it doesn't look like it. Look, guitar pick, kind of, a little bit.
No, I was talking to
Look like a guitar pick, sort of. Teardrop on its side. Okay, that's the RNA polymerase. Okay, that's the RNA polymerase. It'll attach those transcription factors at the TATA box, at that region, and then it's going to start. It's this little red piece here is going to be the start of our mRNA. Again, working from 5. It's attaching or making from the 5 to 3 direction. Okay, always from 5 to 3. Okay. All right. Come on. Go back. There we go. Okay, as RNA polymerase does its job, it moves along the DNA. It untwists the DNA, breaks apart those hydrogen bonds like helicase would do, and it does it really fast. It does it about 10 to 20 bases at a time, but 40 nucleotides per second in eukaryotes. It's fast. And to even speed it up even farther, RNA polymerase usually isn't just one. You'll have multiple RNA polymerases, so you'll have them stacked one behind the other. Just kind of like if you look at the planes at O'Hare, right? Have you ever looked up in the sky and there's like a line of planes? You seen that? It's the same idea with RNA polymerase. So you've got a line of them kind of stacked and ready to get on that runway, to get on that strip and start their job. Okay, nucleotides are added to the 3' end of the RNA molecule. So they're going from 5 and adding on to the 3. Okay, 5 to 3. This is my transcription bubble. This is where it's actively working. And sorry about the. You can kind of see that one, can't you? Wow. What a deal. So it's actively adding them on in that 3', that 3' end. Okay, here's my 5' end, and this is my 3' end. Remember, again, this is my template strand. Yeah, Sophia.
So it only does one side.
And it always only does that one side of the strand. Yeah. So there's going to be a template strand, and there's going to be a coding strand or a non-template. So this says non-template, but it's also coding. Okay. Because if you look at the code, how is this the same or different? Let me clear that. Do it again. My mRNA is exactly the same as the coding strand, except for, the T here is going to have to be replaced. But that's the only thing. Okay? It's the only thing.
Wait, so the red one is the coding.
The top one is going to be the coding strand.
Right.
This guy here.
And then the bottom is the one that
The bottom is going to be the template. So that's template. And this is my mRNA running through it.
that's the one that's
That's the one that's being created. Yeah. All right, cool. Okay. Mechanisms for termination in prokaryotes and eukaryotes are different. And we're not going to spend a lot of time talking about those things. So they're here, but I'm not really going to talk about those. In RNA polymerase, transcription is going to add on a whole bunch of adenines. So this word here, big fancy word, Polyadenylation. Ugh. What's poly mean? More than one, yeah, many. And adenylate. Ada? What does that sound like? Adenine. Yeah, adenine, right? So we've got many adenines that are being added on. That's all it means. Okay. At the very end of the sequence of your mRNA, you're adding on they call it a poly A tail because you're adding on lots and lots of adenines. And next time we talk, I'll talk about why that's done. Okay, why that polyadenine tail is added on. Okay, questions? Yeah, okay. Poly A, yeah. All right, that's the short for that polyadenate. I can't even say it. See, poly A, yeah. So it's just like they add on 10 to, let's say, I guess 10 to 35 is what they're saying. So lots and lots of A's just get added on and on and on. And that's important for the whole process to proceed. If it's not added on, it's not going to happen. Yeah, cool. All right. Any other questions? All right.